Analysis of MSH2 mutation and effect on MMR and HNPCC. I'm Doriana.
My name is Kamaya Watkins.
My name is Deborah Jeffries.
Okay, so the main idea of this experiment is to determine the mutations and the mutations rate of that plays a role in hereditary non-polyposis colorectal cancer, and to also identify the types of mutations in the mutant that can determine the effects that the mutation has on mismatch repair. What is HNPCC, which I stated before, is hereditary non-polyposis colorectal cancer. And it's just an inherited mutation that increases the probability of developing colorectal cancer, and is directly related to MSH genes that occur when cancer is in the body. Screenings for HNPCC can begin as early as 25 years old if an individual has that particular MSH2 mutation, or if the individual has a family history of colon cancer or in the cancer in the lower body. Symptoms may not present themselves, but if they do, it does promote metastasis in the body, which is like cancer can spread to different parts of the body. Or if the person does have symptoms, are typically like rectal cancer. Rectal bleeding, stomach pain, and other cancer-leading symptoms such as weight loss and fatigue. And what distinguishes this particular cancer from other colon cancers is that this cancer occurs at a younger age, such as 25 to 30 years old, compared to other cancers that you can really detect them around 50 to 70 years old. Mismatch repair is just, as we know, a system that recognizes deletions in DNA replication and recombination. So as you see in our model, there's a mismatch DNA and it's cut out and replaced with the correct DNA. And MSH2 is an important in this particular system, and some proteins that are in MMR complex include MLH1 and MLH3, also MSH2, 3, and 6, and PMS1 and 2.
In order to analyze MSH2, a number of experiments were constructed. The first one was plasmid extraction. The objective of this was to isolate the plasmid DNA from 3 E. coli strands, SP1, SP4, and SP0, and measure the concentration and quality of these strands. So first, the bacterial cultures of the 3 E. coli strands were grown in culture overnight. Then the plasmid DNA strands were extracted using the Zippy Mini Prep kit, following the manufacturer's protocol. The DNA samples were then added onto a nanodrop to measure the concentration and ensure the absorbance of the ratios were at 1.8. Next, the identification of sample containing MSH2 insert experiment was conducted. The objective of this was to digest 3 plasmid DNA samples using the restriction enzymes XPAL1 and APAL1, and then to digest the DNA in agarose gel to determine which plasmid DNA contains MSH2 insert. First, the digested and undigested DNA plasmid reactions were prepared. Then these reactions were placed into 1% agarose gel along with 1 kb of DNA ladder. The gel electrophoresis was ran for an hour, and the e-gel image of the agarose gel was released. The results for plasma extraction were SP0 yielded a concentration of 504.2 nanograms per microliter with an absorbance of 1.84. SP1 yielded a concentration of 795.6 nanograms per microliter with an absorbance ratio of 1.87, and SP4 had a concentration of 562.9 nanograms per microliter with a ratio of 1.88. In conclusion, the concentrations and purity of the 3 plasmid samples are acceptable for downstream analysis. For the results of identification of sample containing MSH2 insert, the gel electrophoresis image had an independent variable that were the DNA plasmids, SP0, SP4, and SP1, and the dependent variable was the amount and length of the fragments formed. SP0 digested DNA sample contained 1 fragment in its lane with a size of about 5,000 base pairs, whereas SP1 and 4 digested DNA contained 2 fragments in its lane, about 5,000 base pairs and 4,900 base pairs. SP0 was determined to be the PRS413 because it generated 1 fragment, meaning it was our empty vector, whereas SP1 and SP4 were determined to be the wild type MSH2 and the mutant MSH2 because they generated 2 fragments. The next experiment was the polymerase chain reaction. In order to perform PCR, we did this in order to amplify a region of MSH2 gene that contains the mutant. The plasmid DNA sample was diluted with water to create a diluted plasmid DNA sample. The 3 PCR reactions were prepared and incubated in the thermocycler to begin the PCR program. The data was then observed and recorded. The next experiment was the PCR digestion of the gel electrophoresis. The objective of this was to digest the 3 PCR products using a restriction enzyme BSMA1 to distinguish between mutant MSH2 and wild-type MSH2, and then determine which plasmid contained the mutant MSH2 insert. The 3 PCR products were digested by the restriction enzyme BSMA1. The digested and undigested plasmid DNA were mixed with 6X loading dye and loaded into the 1.5% agarose gel along with the 100 BP DNA ladder. The gel electrophoresis was again ran for 1 hour, and an e-gel image of the agarose gel was released. The results of the PCR digestion and identification of mutant-containing plasmids had an independent variable of DNA plasmids SP1, SP4, and SP0, and a dependent variable that was the amount of length and fragments. SP1 digested and undigested PCR product along with SP4 undigested PCR product contained 1 fragment in its lane with the size of 900 base pairs, whereas SP4 digested PCR product contained 2 fragments in its lane that were the size of about 700 BP and 200 BP. SP4 generated the 2 fragments and was determined to be the mutant MSH2 G704D. G704D MSH2 mutant was identified by entering the mutant SP4 into the APE simulation to find the mutation type. Then the mutation type was found by subtracting location by 11 amino acids, giving us G704D. SP1 was determined to be the wild type MSH2 because it generated 1 fragment.
The second part of the experiment, another part of the experiment was yeast transformation, and the purpose of this particular part was to transform yeast, retransform the yeast reporter strain AGY75 with the mutant MSH2 plasmid, which was our SP4. So the solution contained plasmid DNA, denatured DNA, concentrated SP4 plasmid, and the plate solution along with water. There were 2 transformation reactions created, which was a positive plasmid, which is the experimental one, and it contained plasmid DNA, and also a negative plasmid, which is the control, and it contained water. Those 2 transformation reactions were plated separately on SD his minus, trip minus, and uracil minus plates and incubated upside down at 30 degrees Celsius for 24 to 48 hours. The results of this particular part of the experiment includes the independent variable being the plasmid yeast that is transformed with, and the dependent variable being the number of colonies. And as you can see in our figure, part A shows the negative plasmid and part B shows the positive plasmid. And with this basically saying that the negative plasmid, which is the control, it had no colonies, so the yeast was not transformed with the PMSH2 mutant that we had, because growth of the SD, his minus, strip minus, and uracil minus plate was missing the essential history gene that codes for his amino acids. And then the positive plate, which is the experimental, it had approximately 1,000 to 2,000 growth colonies. So the yeast could grow because the mutant that we had all the essential amino acids, and it coded for the colonies to be grown. So to put this all into conclusion, that this means that the yeast was transformed successfully, and now we know the amount of growth can be observed, or the mutation rate can be observed.
So for the final part of this experiment, the FOA assay, the objective of the FOA assay was to measure the mutation rate of different strains of yeast. And we do this by determining if mismatch repair is working in a transformed sample. So when MMR is working, there's no growth observed on the FOA plate. And if MMR is non-functional, then there is growth observed on the FOA plate. So this concept map shows us how the FOA assay is used to determine the functionality of mismatch repair mechanisms. So when MSH2 is working, MMR repairs the frameshift mutation in G216.5, which keeps UR3 functional, and there will be no growth on the FOA plate. However, when MSH2 is mutated, MMR does not repair the mutation, in GT16.5, which damages URA3, meaning that there will be no toxin that would inhibit growth on the FOA plate. So the independent variable for this FOA assay was the plasmid DNA type and the yeast sample concentration. And the dependent variable was the colony growth and the observed mutation rate. And so there were 9 different yeast suspensions that were plated on the SD, his minus, trip minus, leucine minus, threonine minus, positive uracil, and positive FOA plate. And so the yeast was transformed with either PMSH2, PMSH2 mutant, or the empty vector PRS413. And the yeast strain concentration started at OD equal to 1 and was diluted to OD equal to 0.1 and 0.01. So for the results for this FOA assay, as you can see in this figure here, I highlighted an example of growth. And so the top row is PMSH2, the middle row is the mutant, and the bottom row is the empty vector PRS413. And so we're going to focus on the column all the way to the right, which is at a concentration of OD equal to 1. And so as you can see, the PMSH2 has 1 colony of growth, the mutant MSH2 has 9 colonies of growth, and the RS413 has 5 colonies of growth. And so we can see that this observed data fits the growth pattern, and that there's obviously more growth in the mutant yeast than in the mutant transformed yeast. And so we can see this tells us that the mutant PMSH2 behaves similarly to the empty vector. So the overall objective of this entire experiment was to identify the type of mutation in the mutant MSH2 and to determine the effects that the mutation has on MMR. So to do this, yeast was transformed with PMSH2, and it was shown to have a low mutation rate due to the functioning MMR. And the yeast that was transformed with the empty vector has a high mutation rate due to non-functioning MMR. So through FOA assay, we were able to see that the growth demonstrates a high mutation rate and the no growth demonstrates a low mutation rate. And so we compare the FOA results of the mutant SP4 to the wild type and the empty vector data, which shows us the effect that the mutation has on MMR. So for the connection to Gaming 2007, our mutation present in the mutant SP4 plasmid was G704D, which is equivalent to the Gamma 2007 mutation G693D. And the human cognate, as we can see here, is G674D. And so as we can see in this table to the right here, the G693D mutation belongs to the ATPase category, which has high levels of MSH2, positive subunit interaction, but there's still negative MMR. And so what we can tell from that is that this mutation occurs in the ATP domain of the MSH2 protein, meaning that HTP cannot be hydrolyzed to use for energy.
Real-world impact. G704D is a missense mutation of the MSH2 protein that negatively affects hydrolysis of ATP, meaning it has no energy to form MMR, and it has a higher mutation rate. Knowledge of specific mutation can increase chance of early detection and intervention. Knowledge of how mutation affects MSH2 protein can be used to engineer a cure for HNPCC, such as gene therapy and possibly experimentation with introduction of ATP to damaged cells. And here are the references.